Imagine trying to navigate a really sprawling, hyperdense metropolis.
Okay.
You have this map in your hands, and the data on it is incredibly detailed. Like every single road is perfectly labeled with its street name. Every business is listed.
Right.
But there is a massive catch. The map itself, the actual visual representation of the terrain, is so blurry, you can't actually tell if you're looking at a river, a public park, or, a towering skyscraper.
That sounds like a nightmare.
It really is. You know exactly what the things around you are called, but you have absolutely no idea what the physical landscape looks like where you're currently standing.
Yeah, it creates a complete disconnect. You possess the raw data, the labels, but you lack the physical context required to actually make sense of how everything interacts in space.
Exactly. Which perfectly encapsulates one of the most frustrating hurdles in modern biology. Welcome to a new deep dive, everyone. Today we are exploring a technical report from Nature Cell Biology, published in January 2026, that attempts to fix this exact blurry map problem.
It's a fascinating paper.
It really is. The researchers introduce a computational framework called SpaHDmap. And our mission today is to decode exactly how scientists are using this tool to merge those blurry genetic data sets with crystal clear tissue photographs.
Basically creating a high-definition GPS for human tissue.
Right. Okay, let's unpack this. We already know spatial transcriptomics is this massive leap forward because it gives us the spatial location of gene expression.
Yeah.
But the data we actually get out of it is notoriously messy, right?
Oh, it is a profound mess. And it usually suffers from one of two major extremes, depending on, the specific platform you use.
Right.
So if you are looking at older, widely used platforms like 10x Visium, for example, you run into a severe resolution limit. One single spot on their data grid actually encompasses multiple cells.
Oh, I see.
So you might detect a strong signal for a known cancer gene in that spot, but because the resolution is so coarse, you have no way of knowing if that signal is coming from a malignant tumor cell, an infiltrating immune cell, or just some healthy structural tissue that got caught in the same data bucket.
It's like hearing a conversation in a crowded room, but not knowing which person is actually speaking.
That's a great way to put it. Now, on the other end of the spectrum, we have newer high-resolution technologies like StereoSec or Xenium that can get down to the single-cell or even subcellular level.
Which sounds perfect.
You'd think so. But solving the resolution problem introduces a completely new nightmare. The data becomes incredibly sparse. When researchers simulate this, they use something called a zero-inflated Poisson distribution.
Which sounds like an incredibly complex way of saying the data is full of artificial blank spaces.
Essentially, yes. It means the genes are physically present in the tissue, and they are actively transcribing, but the sequencing machine just missed them. It blinked.
Wow.
If you imagine reading a complex novel where, say, 60% of the words have been randomly erased, you start to understand what researchers are looking at.
So the standard response to this overwhelming, messy, high-dimensional data is dimension reduction. Like, we use math to simplify the data down to its core biological structures.
Right. But the problem is, traditional dimension reduction usually ignores the spatial aspect completely. It just crunches the numbers.
And the spatial methods we do have tend to fall apart when you try to compare multiple different tissue slices at the same time.
Yeah, exactly. Because the physical architecture doesn't perfectly align from one slice of a tumor to the next. What we are really fighting for here is interpretability. If we connect this to the bigger picture, if an algorithm clusters a bunch of data points together and just calls it dimension 1...
That means nothing to a biologist.
Right. It means nothing unless we can definitively link dimension 1 back to a physical reality, like a specific inflammatory pathway or a distinct cellular boundary.
I have to jump in with a fundamental question here, though.
Sure.
If we already possess all this genetic data, even if it is sparse or noisy, why go through the trouble of trying to fuse it with a photograph? Like, we know what the genes do. Shouldn't the chemical signatures be enough to tell us what is happening in the tissue without needing a picture?
If the genetic signal are perfect, maybe. But as we just discussed, the signal is chaotic. Think of gene expression as an invisible chemical frequency.
Right.
If that frequency is weak or constantly dropping out, you are going to misinterpret it. But if you overlay that weak, fragmented chemical signal onto a high-resolution histology image, a physical map showing the actual cellular shapes, the density of the membranes, the architectural scaffolding,
suddenly the noise starts to make sense.
Exactly. The photograph provides the structural ground truth that the genetic data desperately needs, and the genetic data tells you see what those physical structures are actually doing.
It takes 2 slot lenses to bring the image into sharp focus. Okay, so before we move on to how they actually fix this, let's just do a quick recap for you, the listener.
Good idea.
So far, we've learned that spatial transcriptomics gives us gene locations, but it's often blurry or really noisy. Dimension reduction simplifies it, but old methods lose spatial accuracy. So we need a tool that combines the genes with where are the high-res images. So listener, have you ever tried to solve a jigsaw puzzle missing half the pieces? Think about how much easier it would be if you just had the picture on the box to guide you. That's what we're trying to build here.
That's a perfect analogy.
Which brings us to the actual mechanics of SpaHDmap, spatial high-definition embedding mapping. Here's where it gets really interesting. The entire framework is built on a dual-engine mechanism that merges the transcriptomics with high-resolution histology images, like your standard H&E stains. Let's look at the first phase, feature representation.
Right, so this phase is all about prepping the 2 different types of data so they can actually, talk to each other. On the genetic side, SpaHDmap uses non-negative matrix factorization, or NMF.
NMF is a concept we see a lot in data science, but the non-negative part is incredibly crucial when we're talking about biology. Right?
Oh, absolutely.
If you think of a complex data set like a giant pot of chili, NMF is the algorithm that mathematically separates the chili back out into its pure base ingredients, the tomato paste, the beans, the beef. But it is strictly non-negative.
Because biological systems are additive. You cannot have negative gene expression. I mean, a gene is either completely silenced, which is 0, or it is expressing at some positive level.
Right, you can't have negative 3 beans.
Exactly. Many algorithms will use negative numbers to balance their complex mathematical equations, which is fine for finance. But in biology, it creates impossible abstract concepts. By forcing the math to stay non-negative, SpaHDmap ensures that every ingredient it isolates corresponds to a real, physically possible biological part.
Okay, so once we have those purified gene ingredients, the system deploys a graph convolutional network, a GCN. Now, I always think of a GCN as like a sophisticated gossip network.
A gossip network is the perfect analogy. The GCN takes the spots where we actually successfully sequenced genetic data, and it maps them out alongside what their researchers call pseudospots.
Which are just the blank areas on the tissue where the sequencing machine missed the data, right?
Yes, exactly. And the GCN connects all of these spots into a massive web. But it doesn't just connect them randomly. It connects them based on physical proximity and visual similarity.
Okay.
So if a spot with very strong, clear genetic data is sitting right next to a blank pseudo spot, and the system sees that the physical tissue looks identical in both spots, they start to gossip.
Ah, the strong spot shares its genetic profile with the blank spot.
Yes. Effectively teaching its neighbor what genes it should have. This cascades across the entire tissue, denoising the data and filling in the massive gaps.
Wow. And while the genes are busy gossiping and filling in the blanks, the AI is simultaneously analyzing the physical photograph using a UNet.
Right.
Now, a lot of people describe neural networks like this as a microscopic eye, but that doesn't really capture what it's doing. It's acting more like a digital texture cartographer.
Oh, I like that.
Yeah, it isn't just looking at the picture. It is moving pixel by pixel, extracting the mathematical features of the cellular architecture, the exact curvature of a cell wall, the density of the collagen, the FD voids.
So at this point, we have 2 highly refined streams of information. We have the denoised, physically aware gene network and the architectural cartography from the photograph. Phase 2 is where they collide.
The fusion.
SpaHDmap uses an encoder-decoder architecture to fuse them.
The encoder takes the gene map and the image features and mashes them together into a brand new shared high-resolution space. But you can't just combine 2 massive data sets and blindly trust that the resulting map is accurate.
No, you can't. Which is where the brilliance of the decoder comes in. The decoder takes that newly fused high-resolution map and tries to run the whole process in reverse.
Wait, in reverse?
Yeah. It attempts to rebuild the original raw gene expression data and the original histology photograph using nothing but the fused map.
Oh, wow. It is essentially taking a test, grading its own homework, and then recalculating.
Exactly. And that grade is called the reconstruction loss. If the system builds the map, but the decoder's rebuilt photograph looks slightly distorted or the rebuilt gene profile is off, the reconstruction loss is high.
So it knows it messed up.
Right. The algorithm then goes back, adjusts the internal weights of the data, and tries again. It runs this loop thousands of times, constantly balancing the influence of the image and the genes until that reconstruction loss hits absolute minimum.
Okay, that is incredibly clever.
Yay.
Let's do another quick summary for the listener before we look at the real-world results.
Sounds good.
So, SpaHDmap uses NMF to find those interpretable gene ingredients. Then it uses a GCN, the gossip network, to fill in the blank pseudospots.
Yep.
And finally, it uses an encoder-decoder to fuse the genes and images together, constantly checking its accuracy by trying to rebuild the original data.
That's the core engine, yes.
So listener, if you were designing this AI, which would you trust more to guide your map? The physical photo of the tissue or the invisible chemical signals of the genes? Keep that in mind because we're going to answer that shortly.
Oh, we definitely are.
So how does an algorithm that essentially grades its own homework perform on actual complex tissue? The researchers ran it against a battery of current spatial tools, things like Tesla plus NMF, iStar plus NMF.
And it didn't just beat them, it crushed them.
It really did. It exposed some glaring blind spots in how we currently map biology. Look at the adult mouse brain, specifically the coronal section. The brain is famously structured, with highly distinct layers in areas like the cortex and hippocampus.
Right, and SpaHDmap mapped all the known layers perfectly. But the real breakthrough was finding something the researchers labeled cluster C9.
Cluster C9, what exactly is that?
Cluster C9 represents an ultra-thin biological structure called the leptomeninges and the glia limitans. Think of it as a microscopic, incredibly delicate biological saran wrap that encases the brain.
And the other computational models completely missed it.
Completely. Their resolution was either too blurry or they couldn't separate the noise, so the faint chemical signal of this ultra-thin membrane just got entirely swallowed up by the massive, loud signals of the brain tissue right next to it.
Which is a catastrophic failure if you were a researcher, right?
Absolutely. The meninges are not just physical shock absorbers for the brain. They are a critical, highly active hub for central nervous system immunity and homeostasis. Immune cells constantly patrol this exact barrier.
Wow.
If you were developing treatments for neuroinflammation or researching how Alzheimer's disease progresses or studying meningitis, you absolutely must be able to see what is happening in that delicate layer. SpaHDmap isolated it flawlessly where others saw nothing.
Finding a healthy, intact anatomical barrier is impressive. But what happens when the cellular architecture is actively being destroyed and mutated by cancer?
That's the real test.
The researchers tested this on medulloblastoma, which is a very complex type of brain tumor, using human tumors grown in mice PDX models. They had 4 samples. 2 were treated with a cancer drug called palbociclib, and 2 were untreated controls.
And this specific test highlights what is arguably the biggest nightmare in computational biology: batch effects.
So what does this all mean? We hear about batch effects constantly, but the way they ruin data is wild. You could run a tumor sample on a Tuesday, and I don't know, the humidity in the lab is a bit high.
Yeah, exactly.
You run the next sample on Thursday with a different lab technician and a slightly older batch of chemicals. The underlying biology of those 2 tumors could be identical, but the sequencing math will look different purely because of how they were handled.
And standard AI models get completely derailed by this. If you run a standard factorization model on this medulloblastoma data, the algorithm groups the cells based on which day the lab test was run, rather than grouping them by whether they are cancerous or healthy.
Oh, wow. So the technical noise overpowers the biological signal.
Yes. But SpaHDmap has a built-in mechanism to strip that technical noise away. The researchers ran statistical mixing tests to prove it. While the older models kept the lab batches mathematically separated, SpaHDmap aligned the samples perfectly.
And once it stripped away the artificial batch effects, the true biological reality emerged.
Right. It allowed researchers to differentiate between 2 very specific genetic behaviors happening simultaneously. It could clearly identify the sonic hedgehog or SHH, transcriptional signatures.
Which represent the fundamental cancerous identity of these specific tumor cells.
Exactly. And at the same time, it could map the E2F target genes, which represented the cell's active reaction to the palbociclib drug treatment.
It separated the identity of the cancer from its current state of being medicated.
Hmm.
That is a level of clarity you need if you want to know if a drug is actually working on a cellular level.
It's vital.
But I have to push back here. Everything we've discussed, assumes the data going into the system is at least decent. We know laboratory runs fail. What if the histology image is a blurry mess? What if half the genes fail to sequence? Does this algorithm just start hallucinating structures that aren't there?
That is the exact vulnerability the researchers wanted to test. They intentionally sabotaged their own datasets to find the breaking point of the AI.
Let's do a quick recap before we get into the sabotage.
Oh.
So SpaHDmap finds structures other tools miss, like that ultra-thin C9 meninges layer in the brain. It can analyze multiple samples at once, erasing those technical Tuesday versus Thursday batch effects.
Right.
And it successfully distinguishes between different tumor states in treated versus untreated samples. So listener, think about medical testing for a second. How important is it that an algorithm can tell the difference between a biological change from a drug versus a glitch from the lab equipment? It's literally a matter of life and death.
It really is.
Okay, so back to the sabotage.
Yeah.
They didn't just tweak the data, they took a sledgehammer to it.
They did.
They restricted the algorithm to only look at 500 genes. They intentionally erased 60% of the data to create massive artificial gaps. They injected mathematical noise into the equations, and they degraded the high-resolution tissue photographs down to 164th of their original quality.
And despite all of that degradation, SpaHDmap's mapping accuracy remained remarkably stable. When they looked at the dentate gyrus in the brain, the algorithm still accurately mapped the delicate spatial architecture, even with 60% of the genetic signals completely missing and a highly pixelated image.
It didn't hallucinate.
No, it didn't. It relied on the deep patterns it had learned to bridge the gaps.
Which leads directly into what is easily the most mind-bending part of this entire paper, Case Study 3. They ran a dataset for human colorectal cancer, sequenced by StereoSeq, but there was a massive piece missing.
Yeah, there was no histology image.
None. No photograph of the physical tissue existed for this specific dataset.
Considering the entire premise of SpaHDmap is fusing the chemical gene map with the physical tissue photograph, this should have been a hard stop. The system shouldn't work.
Right.
But the researchers utilized a fascinating workaround. They instructed the algorithm to generate a pseudo-image directly from the chaotic, noisy genetic subcellular data itself.
It essentially used that gossip network we talked about earlier to draw a picture of where the cells should physically be based on their chemical similarities. It dreamed up a fake photograph purely from invisible chemical signals.
And incredibly, using nothing but that AI-generated pseudo-image, SpaHDmap correctly distinguished the intricate boundaries of the colorectal tumor subclusters from the stroma, which is the supportive tissue surrounding the cancer.
While the competing methods completely fell apart.
Oh, they failed miserably. Because the competing models were rigidly designed to only read actual physical photographs, they couldn't process the pseudo-image. They ended up misclassifying massive regions of highly malignant colorectal tumor as harmless, healthy stroma.
Misclassifying a tumor as healthy tissue is the ultimate failure for a diagnostic tool.
It's the worst-case scenario.
But the fact that SpaHDmap could adapt so flawlessly brings up an interesting mechanical question. How does the AI actually know when to trust the photograph and when to ignore the photograph and trust the genetic data instead?
What's fascinating here is that the scientists actually control that balance using a tuning dial called the lambda parameter.
The lambda dial.
Yes. So when they are feeding the system an H&E stain image, which provides incredibly rich, distinct architectural details of the cell walls and structures, they set lambda low to 0.33. This mathematical weight tells the algorithm the picture is highly reliable, lean heavily on the visual data to guide the map.
But not all images are that detailed. Like, if they use an IHC stain, which uses antibodies to highlight specific proteins but washes out a lot of the overall structural background, the picture isn't as helpful.
Exactly. So for IHC images, they turn the lambda dial up to 0.9. This shifts the mathematical weight, telling the algorithm to deprioritize the blurry image and place almost all of its trust in the genetic sequencing data instead.
Oh, that makes so much sense.
This lambda parameter is what makes the system so incredibly robust. It doesn't blindly apply one rule to every data set. It adapts to the quality of the information it is given.
Okay, let's do one final recap before we wrap this up.
Before.
The system is highly robust against noise, missing data, and low-res inputs. It uses that lambda dial to adjust how much it trusts the image versus the genes. And it can even generate its own pseudo-images to find tumors when no physical photo exists.
A very solid summary.
So listener, does it make you nervous or excited that AI can accurately map cancer using a pseudo-image it dreamed up from raw genetic noise? It's a lot to process.
It really is. This raises an important question about how much we should rely on algorithm-generated pseudo-images versus physical reality moving forward.
SpaHDmap represents a fundamental shift in how we approach biology. We are moving away from treating a tissue sample as either just a visual photograph or a spreadsheet of chemicals, and finally merging them into a single, cohesive reality.
Yeah.
For anyone involved in medical research, the implications are profound. When we look at the future of targeted cancer therapies, the ability to map the exact boundary where a tumor ends and healthy tissue begins, even when the data is noisy and degraded, means treatments can become fiercely precise.
We are taking that incredibly blurry map of the city and finally bringing it into razor-sharp focus. We can finally see the street names, the buildings, and the traffic flowing between them, all in real time, without losing the context of the wider neighborhood.
And that leads to a fascinating final thought to consider. We just explored how an algorithm can generate a highly accurate, predictive pseudo-image of a tumor from pure genetic noise, successfully mapping cancer without a physical photograph.
Right.
If computational models are already this adept at predicting physical architecture from chemical signals, are we rapidly approaching a threshold where physical biopsies and traditional optical microscopes become entirely obsolete?
Wow.
Will we soon reach a point where we simply sequence a single drop of fluid and allow AI to perfectly render the complete microscopic reality of our own tissue? Thank you for joining us on this deep dive. Keep questioning the map, and we will see you next time.